So far, you have already heard about genomic structural variation in Lior Pachter's talk and also Mike Schatz's talks. But now you're going to know more in detail. So when we are talking about genetic variation, it comes in different sizes. So the most commonly known ones and the most assessed ones are single nucleotide polymorphisms, which are in short called SNPs. And there can be longer polymorphisms, which are called small indels or microsatellite expansions or contractions, up to 50 base pairs. And now the structural variants are those variants that affect more than 50 base pairs. So the traditional definition was about a KB, but with the introduction of next-gen sequencing, with the 1,000 Genomes project, now we are talking about down to 50 base pairs. And these can be deletions of DNA, insertions of either novel sequence or copy-paste events, which are mobile element transpositions, segmental duplications. So this is duplication of larger segments of DNA with sequence similarity more than 90% between paralogs, and also inversions of DNA and translocations from one chromosome into another. And much larger changes are the chromosomal changes. So these are the ones that are seen using advanced microscopes. The most famous example is the 21 trisomy, which causes Down syndrome. And in general, those ones, the deletions, insertions, and segmental duplications, are also called CNVs, which stand for copy number variants, because they change the amount of DNA in the genome. And when we are talking about structural variation, although the number is smaller, so we see about 2,000 to 500 structural variants depending on the size and the type of the structural variant in a human genome, they affect much of the DNA. So with SNPs, we usually have 3 to 4 million SNPs, which changes only 3 to 4 million characters. But structural variants, because they are larger, they affect up to 20 megabases of DNA. In normal, I mean non-disease human individuals. So this is the map of human genome structural variation created from the genomes of 9 people back in 2008. And I was a postdoc in Evan Eichler's lab back at that time in University of Washington. And Jeff Kidd did this $10 million project and analyzed 9 genomes, and he came up with this genome structural variation map for insertions, deletions, and inversions. And you can see that they are pretty much everywhere, although there are some hotspots here. I believe this is the HLA region, and some other hotspots in chromosome 17. So why do we care about the structural variants? One thing, the loss of CNVs are already associated with human disease. So again, this is old data. If you remember from Dana Pierre's talk, she was talking about microarrays. So this is back in microarrays time. What Andrew Sharp and others did was look at what we call CNV hotspots. Basically, these are those regions that are flanked with segmental duplications of size 10 kb. And these duplications are within 50 kb to 5 megabases. And then he interrogated those regions using microarray experiments, and then they associated some mental illnesses as far and also autism spectrums and schizophrenia and so on with those CNVs. So what happens is when we have a duplication such as this one, this may cause unequal crossover. So this is the hypothesis that defined the CNV hotspots. This unequal crossover happens because those paralogs are very similar to each other. And then during the meiosis, you have 2 different gametes where you have an extra duplication of this green region and also the region in between those duplications, and another gamete losing 1 copy of the duplication and also this ABC region in between. And if they hit some important genes, then they may cause diseases. So again, when we are talking about these CNV hotspots, we can find them throughout the genome looking at the segmental duplications. And this is the map of human segmental duplications in build 36, I believe. So this slide is a bit old. And these blue lines basically show the copies of different duplications. And this is only the intrachromosomal ones. And based on the CNV hotspot definition, so those gold bars are the regions where we expect more CNVs. And so far, these diseases, and so this is actually slides from back 2002, a lot of diseases have been associated with CNVs. So again, this is intrachromosomal hotspot, intrachromosomal duplications. And if we also look at the interchromosomal duplications, we have a much complicated map. So those duplications are all over our genome, which covers about 5% of the genome. And about 52,000 pairs of segmental duplications have been identified. So if it is intrachromosomal duplication, it is probably a CNV hotspot. And if it is interchromosomal duplication, this may be translocation hotspots. Another reason why we call about copy number variance is the copy number polymorphism is also associated with disease. So this time we are not talking about deletions, we are talking about duplications. Right? Here are some diseases that have been associated. For example, an decrease in the copy number of C4a, C4b gene is associated with lupus, and an increase in defensin gene family is associated with psoriasis. So the defensin genes are already duplicated genes, so they are within those segmental duplications. And if we have an extra copy, it is likely that the person is going to get psoriasis or susceptible to psoriasis. And in the same gene, again, these are duplicated genes. If a person is missing a copy, this condition is also associated with Crohn's disease. And we have other examples, such as the decrease in CCR3L1 gene increases HIV susceptibility, and increase in the CCR3L1 gene copy number shows better resistance to HIV. So again, previously this work has been array-based, but with the introduction of next-gen sequencing technologies back in 2006, 2008, we started to look at those regions using these new tools. And it actually gave us better power because the microarray technologies are biased against duplicated regions, because we are only looking at the log2 ratio of the signal intensity. So that means if we have 7 copies versus 8 copies in one individual and the other, then you are looking at a logarithm of 7 divided by 8, which is very similar, which is very close to the background noise. When I was a postdoc at University of Washington, my initial project was actually taking a leverage of those next-gen sequencing technologies to assess the copy, the content, and the structural of segmental duplications. So let's step back a little bit and talk about those structural variation classes. Again, these may be caused by segmental duplications or even large common repeats such as ALU or L1 elements. And it can be deletion of some DNA, which is associated with autism, mental retardation, and Crohn's disease. Novel sequence insertion, so this is the sequence that are not represented within the reference genome. Mobile element insertion, so these are the copy-paste events of known sequences such as ALU, L1, and SVA, which is associated with hemophilia, for example. Or duplications in tandem, or duplications in interspersed structure, which is associated with schizophrenia and psoriasis. And there are also inversions of DNA and translocations of DNA, which is associated with leukemia. And this is very common in cancers. Again, these are called the copy number variants because they change the amount of DNA. And those two inversions and translocations are called balanced rearrangements because they usually do not change the amount of DNA. So again, historical lesson here. The old work was based on hybridization method. Back in 2004, I believe this was the first CNV paper. They used back arrays to find some large deletions. And then in 2008, SNP microarrays were used, and then array CGH technology was used. And another method called optical mapping was also developed in 2010. So, and these are all wet lab-based methods, basically. And now we have the bio-nanotechnology, which is an easier and more cost-effective version of the optical mapping technology developed back in 2010. With the sequencing data, the first algorithm was introduced by Jeff Bailey in 2002. It was a redep-based method, which I'm going to talk more in detail. And then in 2005, Tuzun used the phosmid and sequence pair approach, and then Jeff Kidd added 8 more genomes to this study. And Sanger sequencing data was used by Ryan Mills to mainly assess indel polymorphism. And then with the introduction of, so these are all old Sanger sequencing-based methods. So here each genome costs about $1 million or so. And then with the introduction of next-gen sequencing technologies, it was a boom in algorithm development, and many different algorithms have been developed since 2007. And as you know, the 1,000 Genomes project was the big project that used the next-gen sequencing to assess all forms of genomic variation. So there are these different methods, but what do they give us? So is any one of them better than the other? The answer is no. So this is the gains and losses. So here the gains are insertions and duplications, and losses are the deletions that are larger than 5 kb detected in the same 5 individuals using 3 different technologies. And these are all validated real deletions. We are not talking about any false positives here. And if you look at the phosmid clone and sequence pair data from Jeff Kidd back in 2008, it can find 800 more structural variants that no other technology could find. But similarly, ultra-dense array CGH technology could find about 600 that nobody can detect. Right? And as you can see, the intersection between different technologies are pretty small, especially this region. Right? So none of those technologies are better than the other, but today, obviously, we are only using the next-gen sequence technologies. It's not better than the others either, but it is way cheaper. So let's talk about this old technology first. So this was the clone and sequencing. We first start with the DNA of the sample that we want to analyze. We shear it into large segments, and then we clone them into phosmids or back vectors. And this is bacterial vector sequence. And we have the human DNA here. We put the human DNA here. And then we give this vector back to the bacteria, and then it copies it for us. And after that, we already know those locations where we inserted our human DNA. And then we cut it again. We have the human DNA back. And then we sequence from left and right. And again, this is all technology. So we used Sanger sequencing, and we got about 1,000 base pairs from each side. So this is the method that Eray Tuzun used and then Jeff Kidd used back in 2005 and 2008. So this is expensive, more than $1 million per genome. It takes a few months to generate this data. And it is very labor intensive as well, but it is computationally easier because we have long sequences and we have long molecules to interrogate. So now what we do is, obviously, whole genome high throughput sequencing. So now instead of cloning, we just have the test genome. We shear it into smaller segments. And then we either use Illumina to generate paired-end sequencing, which gives us similar data type, but much shorter, obviously. Or we use some PacBio or Oxford nanopore technologies to generate single-end sequencing data. And then we map them to the reference genome, and we look for changes in the mapping properties than expected. So there are basically 4 different sequence signatures that we can use to assess structural variation. The first one is read pair analysis. So this is the same methodology that Tuzun and Kidd used. So we have this molecule. We sequence from the prefix and the suffix. And we know the size of this molecule approximately. For Illumina, this is usually 400, 500 base pairs. And then when we map them back to the genome, if their map location is separated much more than the expected size, then this likely shows you a deletion. And you can come up with similar formulations for insertions, inversions, transpositions, and so on. So this is a very, very commonly used approach, but the size of the variance that you can detect and the resolution of the actual breakpoints depend on the insert size and the read length. The second method is read depth analysis. So this is from Jeff Bailey back in 2002, again using Sanger sequencing. But again, we apply it to high throughput next-gen sequencing. We basically just map the reads, and then we just count the reads across the genome. And then if we see a depletion, it is likely a deletion event. Or if we see an increase in read depth in a region, it likely shows us duplication. So we also can use split read analysis. So here, if our read is just spanning the breakpoint, then when we map that read back to the genome, it will probably split into two pieces, which will tell us that there is a structural variant and what type of that structural variant. And we can also do assembly. So this is, again, back to Mike Schatz's talk. We start with the reads, we assemble, we get larger contigs or scaffolds, and then map this to the reference genome and look for differences. So there are lots of different structural variation discovery tools. Another one pops up pretty much every day. And these are some of them that used read pair, read depth, split reads, and also more recent ones use a combination of those sequencing signatures, such as using both read pair and split reads, or using read pair, split reads, and read depth. Right? And they can detect different sizes or different types of structural variants. So again, these tools, most of those tools were developed within the 1,000 Genomes project, and in the first pilot phase, 19 different algorithms were used to assess structural variation in 1,000 Genomes pilot. And again, they all use different sequence signatures. They use different platforms. So back in that time, there was the solid platform. There was the 454 platform. Now, as you know, Illumina is the dominant one. And they could detect different types of variants in different sizes with different false discovery rates and different false negative rates. Right? So now we already know that no technology is superior than the other. Is any method superior than others? Again, the answer is no. So looking at those 14 of those 19 algorithms, so excluding the assembly-based ones, if we look at those validated deletions again, we still see differences in detection power. So 6 algorithms that used read pair method is here. 4 algorithms that used read depth is here. And 4 algorithms that used split read is here. And again, we see very little intersection between those two. And I have heard before that people are referring to this slide as the terrible Venn diagram. So again, back to University of Washington. So when we started dealing with this data for 1,000 Genomes project, we started looking at the challenges in using next-gen sequencing data to assess structural variation. So the first challenge is to deal with the short reads. So when we have the short reads, today we have 150, more or less. Back at that time, we had 36. So even with the 150, we still have lots of mapping artifacts because the human genome, half of the human genome is repetitive. Those sequences can map to many different places with pretty much with the same edit distances. So what are we going to do? We can either discard the non-uniquely mapping reads, which is going to cause us lose sensitivity in detection, or we can try to utilize all the information to gain higher sensitivity with the cost of less specificity. So we selected the second approach because in our lab, our primary focus was duplications and the variants that are flanked with duplications. So we first started implementing a read mapper that returns all possible map locations. And then looking at the mapping information that this mapper generates, we develop a read-depth-based method, and then read pair, assembly, and split read-based methods to detect different types and sizes of structural variants. But now, as of 2 months ago, I believe, we have a newer algorithm which I call TARDIS. So if there are any Doctor Who fans here, they will know that it is bigger in the inside, so all of those algorithms are inside. This one is not yet, it will be soon. So how do we do the read depth analysis? Okay, so next slide is costly. Okay, so the read depth analysis was again developed first in 2002. So if we have this sequence to test, let's say we have this DNA, and then we have all the reads generated from this DNA randomly. And let's say this region is deleted, so there is maybe heterozygous deletion. This black region is normal, and the red region is duplicated. So what will happen is, when we map those reads, it will take a while. Done? No. So when we map those reads, we are going to see a depletion, maybe no reads mapping into the blue region, and some reads mapping to the black, and increased number of reads mapping to the red. And then we count them using a sliding window. We can now identify the regions that are likely deleted and likely duplicated. So why we do use the multiple mapping data, multiple mapping locations? Again, this is specifically for duplications, because if we have this genome and we have this duplication, so this region duplication, this region duplicated, and then we have the reads, if we map them to unique mapping locations, everything will just look normal. You're not going to see any increase in copy, unless we have a third copy of this one. So if this is already duplicated in the reference genome, you're not going to see a difference. So if you have a third copy, then you're going to see a difference in those intervals. Or if you use multiple mapping, then you are going to see all regions of duplication, regardless if it is duplicated in the reference or not. So when we are looking at the read depths and when we are counting, so we also have to take into account that the read depth distribution is not uniform. It's not even Poisson. There are biases against high and low GC content regions. And these are 3 different sequence data sets from the 1,000 Genomes project. And then if we plot the number of reads versus the average GC bins, sometimes you see a pretty much decent distribution with some noise here. Sometimes you see a distribution like this. Sometimes you see a distribution like this. So you have to fix these biases per each sample. So what we first did was to discover the segmental duplications in those genomes. So we applied some next generation sequencing GC error correction, GC normalization, based on the lowest smoothing technique. And we also removed the common repeats from consideration. And then at the end, we were able to predict the copy numbers in 1 KB non-overlapping windows. And then we could assign integer copy numbers to duplicated genes. So this was something that we were not able to do before with the microarray technologies, but the next-gen technologies gave us this power. And when we look at different genomes, this is how it looks like. So the green regions are the unique regions. So these are copy number 2, because each chromosome has 2 copies. And the red regions are the regions of segmental duplication. And we can see differences in different individuals. So for example, this region was not duplicated in Craig Venter's genome, but it was duplicated in James Watson, and also those 3 genomes from 1,000 Genomes project. And this region was not duplicated in this and this individual, but it was duplicated in the last individual, and this is actually a family. So another method that uses similar signature is CNVinator. Again, this was developed within the 1,000 Genomes project by Alex Ebizov. So now this one uses only unique mappings. So it performs better for deletions. and maybe some smaller duplications as well. And looking at the unique mappings and the read counts, they apply the mean shift technique to partition the read depth and then assess the regions of duplications and deletions. It is also possible to differentiate between the paralogous genes with next-gen sequencing. So for example, here we have two different gene families, CFHR, which has 5 copies in the reference genome. And these are highly identical to each other. So if we just look at the mean copy number, we can just say this person has 3 versus this person has 5. But if you want to know which of those copies is depleted or has an extra copy, we look at those variants that we call single unique identifiers. And another example here is the opsin gene family. So for example, this genome, this person from Nigeria is missing the middle wave opsin gene. Okay. So this is the single unique nucleotide identifiers, or SUNs in short. Let's say we have 6 copies of a gene. We align them. We do multiple sequence alignment. And then we just look at column by column, and then we identify those positions that is unique in one of those copies. So for example, this C is unique in copy 2, and in the same region, they are all T's, I believe. Right. And then if we look at only those reads that map to those unique identifiers, now we can assign a paralog-specific copy number to duplicated genes, which here is exemplified by the emblaze gene family, which has 6 different copies. And these are the copy numbers across 159 individuals from different ethnicities. And you can see that some genes are missing copies in some individuals, and some genes have extra copies in other individuals. So the second technique is read pair analysis. Again, we already talked about this. So if the reads are mapping in an unexpected way, it signals a structural variant. And we can formulate deletions, novel sequence insertions, transposon insertions, inversions, and duplications. So there are different algorithms that use this technique. The algorithm that we developed was VariationHunter, and it was using all possible map locations, and it formulates the problem in as a maximum parsimony approach, and then it tries to find the optimal number of structural variants using a reduction and an approximate solution to set cover. And there are similar approaches such as Clever, Hydra, Breakdancer, and a geometric formulation of using the same signature is GASV. So now this depends on the span size. So this doesn't really depend on the GC percent, other than the fact that higher GC regions mean that there won't be any reads coming from that, right? But it also depends at the span size distribution, so the sizes of those molecules. So this is a good data set. This is old, but it's a good data set. So the average size of those molecules is 208 in this example. And if we just plot it, it is nice and tight. And if we see those read mapping that are smaller than this span, we can call them as insertions, or if they are larger, then we can call them as deletions. And we also call those read pairs that map within this red region as concordance, and those that are outside as discordance. So again, this is a good one, but there are bad examples, right? So sometimes we get span size distributions like this, and sometimes even worse. Sometimes we even have bimodal distributions. Since we are running out of time, I'll just skip those very, very quickly. Basically, this is the mathematical model of VariationHunter. So this is an example for insertions. Again, let's say we have the DNA here, and we have this sequence inserted, and we have this paired and reads coming from the same molecule. Let's say this molecule size is about 500. When we map them, they are going to map closer to each other than expected. And then we just define those paired and clusters with predicted breakpoints for the insertion. So the insertion should be within here. And the map locations of those read pairs and the orientations of those read pairs. And then we come up with this tuple. And then after this, we can just cluster those that agree with each other. Let's skip that one as well. So we just cluster those that agree with each other for the same type of structural variant and the same size of structural variant. And we remove those that are in contradiction. For example, here, this would be in contradiction. Because here the red and the green are agreeing on the location of the insertion, but the blue one is just violating this condition. And then we find those that agree with each other, we cluster them, and then we reduce this problem into set cover, and we find an approximate solution, which is going to give us the potential locations for the structural variants. So the third methodology is the split read analysis. Again, as I told before, if we have paired end reads, so we can also apply this one to PacBio or nanopore data because they are single end already. They are not split. They are not paired. So when we have pairs, maybe one read is going to map normally. The other one is going to be split into two pieces. And depending on how it splits, or if we have PacBio or nanopore, we are going to have one read split into two pieces. Depending on their split, we can come up with formulations for deletions, insertions, inversions, transposons, and duplications. Again, there are many different methods that use split reads. The first one was Pindel back in 2009, and now then newer ones started to show up, such as Schrick, and then split read, and for RNA alternative splicing. So this is not really for structural variation, but the formulations are a bit similar. For alternative splicing detection, Top Hat was also using the split read analysis methods. So the last example would be the local assembly. So we first developed this NovelSeq back in 2010. We have a newer version called Pamir, and there is a Popins tool from Iceland, from Paul Malstedt's group. And when we have a novel sequence insertion and we have short read pairs, they are not going to span this insertion, but we are going to see from left and right of this novel sequence insertion, we are going to see what we call one-end anchored read pairs. And here, those reads that are coming outside the insertion are going to map normally from the left and from the right, but the reads that are coming from inside the insertion are going to be unmapped. So that's why they are called one-end anchored reads. And there will also be the orphan read pairs that come, both reads are coming from the insertion, and they are not going to map anywhere. So we take those, we cluster those one-end anchored reads, and then we infer the potential location of the novel sequence insertion, and then we assemble the rest. Right? So we assemble the orphans, and then we do some contamination filter, because when you are sequencing, you are not only sequencing the human genome, you're also sequencing anything in the blood. There can be E. coli, there can be yeast in the air. And when I look at some data from 2011 from another group, I even found rice in the sequenced genome. So there can be different genomes in your sample. And then we assemble the one-end anchored parts, and we assemble the orphan parts, and then we basically just match them using an algorithm for maximum bipartite matching. Right? So let's revisit the structural variation and next-gen sequencing. So far, everything that I was talking about was for historical and for teaching purposes, really. But now we have newer technologies and more exciting technologies that give us additional power in structural variation assessment. So again, there are different challenges in SV detection using NGS. Some of them are issues related to genome structure. There is high sequence complexity at the breakpoints of structural variants. That means there are common repeats in the flanks or segmental duplications in the flanks. And there are also sequencing issues. So we have short reads, we have erroneous reads, we have GC bias, and we have short fragments. Right? So what are we going to do? Let's look at old versus now or new, and the newer technologies. So the old ones was cloud-based, So the good thing was it provides long-range information and also long reads, but they are slow and they are expensive. So the now technology is NGS. It is fast and inexpensive, but it has short-range information, and there are errors and repeats are problems. So what if we combine the best of those two worlds? So we can do this using either pooled clone sequencing or linked read sequencing. So pooled clone sequencing was introduced back in 2011, originally developed for long-range haplotype phasing, but we could leverage the same data for structural variation discovery. And linked read sequencing is, again, an easier way of doing pretty much the same analysis. So just like the optical mapping back in 2010 and then Bionano today, so this is back in 2011, and we have 10x genomics today. So this is what we mean with long-range information. So again, we have, so this is the pooled clone sequencing version. So this is relatively harder version. We have the genome. We chop it into large fragments. Again, we clone them using BECs or phosmets, and then we dilute them into a number of pools. So with random dilution, there is a very low chance of overlap between those molecules when mapped to the reference genome, and each pool covers about 3% of the genome in this method, right? So 3% of the genome total. So there is a very, very low chance of overlap within a pool. And then we barcode those pools, and then we apply Lumina sequencing, and then we generate some data, and then we can just assign every read to a pool. And here, each pool is going to include a number of about 300 or so back molecules. Now, we still have Illumina sequencing, so the sequencing part is cheap, right? This is kind of labor intensive, but at least we are not dealing with $1 million genomes anymore. Similarly, 10x is giving us similar data somewhat. So again, we start with the genome. So we again have large segments, but we are not cloning this time. So the cloning part is labor intensive. It takes time. So we are not cloning this time. But again, we are dividing into pools. So there is fancier slides from 10x genomics, but I skipped those because it can be confusing. But basically, we are again dividing into pools. Now we have a lot more pools, and now we barcode and then sequence. But the change here is instead of about 3-4x coverage with pooled clone sequencing, here we have 0.1x coverage per pool. Okay. So the differences between pooled clone sequencing and 10x are as follows. With pooled clone sequencing, we use cloning, and we can have 40 kb or 150 kb clones depending on the vector that we use. In linked read sequencing, we have average 50 kb. I heard yesterday that now you can get about 200 kb molecules with 10x. So with pooled clone, it requires cloning, but with linked read sequencing, there is no cloning and everything is automated. With pooled clone sequencing, you get decent coverage, about 3x coverage. With linked read sequencing, you get about 0.1x coverage per molecule. You have about 300 pools in pooled clone sequencing, and you have up to 4 million pools in 10x genomics. Right? So although this cartoon looks similar, the data is actually somewhat different. Right? So now, looking at this data, what we first tried to do was to identify inversions. So because deletions is relatively a solved problem, because it is, among all those structural variants, it is the easiest one to detect, because you can also use read depth information. But the inversions have not been attacked before. There is some development within 1,000 Genomes project, but the false discovery rate was about 70%. And it was only limited to short inversions. So what we want to do is to look at large inversions, megabase scale inversions. So let's say we have this reference genome, and then we have this segment. And then in the sample that we sequence, this segment is flipped. So this part is inverted. And then let's say we have sequences coming from this genome, and we have some read pairs coming outside the inversion. some read pairs coming inside the inversion, and some are spanning the breakpoints. And what will happen is these are going to just map normally. This is also going to map normally. When you look at the mapping, you're not going to see the change in the orientation. But those are going to be split. So this will be similar to the split read analysis, but in this case, we are now dealing with larger molecules, right? And we are going to just look at the full mapping of those read pairs. And this will also be split, and this will also be an additional read pair information. So that signature is going to be for the typical read pair. So when we started looking at this as a responsible PI, I started to think about algorithm names, and I couldn't find any. So I tweeted about it, right? I was asking for help, and then Lior came. And Lior said, just find somebody else's algorithm and name your algorithm the same. And then I said, OK, Callisto then. Right? He actually liked it. I mean, he hit the like button. But we didn't call it Callisto. Instead, we called it VALOR, so variation using long-range information. So this is the signature that we used for inversion. So we can, we have the same formulation for pooled clone sequencing and 10x. The only thing that changes is the size of the molecules that we have to deal with and the coverage, obviously. And again, let's say this is the inverted haplotype in the sample. So if we have molecules spanning the breakpoints, they will be split into two. And since those molecules are very, very unlikely to be in the same pool, right? So those molecules should not be in the same pool. And again, it's very unlikely in the same pool. And let's say this is in the green pool, this is in the blue pool. And we are going to see splits in different pools. Right? And so this will be our split molecule signature or split clone signature for pooled clone sequencing. And in addition, we have the traditional read pair signature as well. So this read pair is spanning this breakpoint, and this read pair is spanning this breakpoint, and they will also map in different locations. So when we have those split molecules, again, obviously, we are going to cluster them, just like we did for read pair analysis. So let's say, again, this is our inverted haplotype. We have in red pool, we have this split clone. So we call it a split clone when the size of this inferred clone, so this is inferred by just looking at the mappings within windows and then cluster those that are in close proximity to each other in the same pool. And we already know the size of each molecule, which should be about 50 kb for 10x and about 150 kb for back pooled clones. But if this size is much smaller than that, but if there is another small region somewhere close by within the limitations of the inversion that you want to detect, let's say you are looking up to 10 megabases of inversion. So if there is another region in the same pool, again, smaller than expected, but if you just add them up, you are going to get somewhat the expected size. We call those as split clones. Obviously, there are lots of false split clones in there. That's why we do clustering. And then we have additional information from the blue pool. And then we cluster those. If they agree, we allow for some gaps and overlaps because of the repeats in these regions. So there will be mapping ambiguities. And then we call this as a potential split clone inversion. And in actually, we expect more than one reds and more than one blues. So obviously, they will be green and gray, whatever. And then basically, the algorithm is we first start reconstructing those molecules by merging the read elements in close proximity within the same barcode or within the same pool. And now we find pairs of those recovered molecules with the same barcode that are shorter than the average size, but when they are added up with another short one in close proximity, it gives you the expected length. And then we find pairs of those compatible split molecules. And then we denote those pairs of split molecules as nodes in a graph. And then if we have additional pairs of split molecules that also agree with those, we put an edge between them, we get a graph. And then on this graph, we find maximal quasi-clicks in this graph. So these are not full clicks, but they are approximate clicks. And then for each click, we also look at the read pair information. We score them up, and then we come up with a potential list of inversions. So we first applied this to pooled clone sequencing data generated from the genome of NA12878. And it had about 300 backpools. And basically, we look at inversions that are larger than 1 megabase up to 10 megabases. And within this size range, there were only 3 in the literature that were previously validated in the wet lab. We could find all 3 of them. And we could also find another one that we validated ourselves, so in chromosome 16. And this is the entire set of our predictions that we could design some validation experiment. So we applied the same methodology to 10x genomics. So now we had 3 genomes. One of those genomes was the same. And we also had her husband and her son. With the 10x genomics data, we just changed our expected molecule size, obviously. And then this is, again, the entire set of our predictions. And again, those 3 that were validated before we could find. So we also could find the one that we found in the pooled clone sequencing data, again, using the 10x genomics. And a friendly reviewer just told us that this is also real. So in chromosome Y. Except that the father is missing it, so this should be a false negative. Or he's not the father. OK, and finally, this is our visualization of this inversion. So these are the split clones from one side, and these are the split clones from the other side. So you can see, we see multiple split clones signaling the same inversion. And these are the read pairs in this inversion. And this is the fluorescent in situ hybridization validation. And we can talk about how this works if you're interested. And we can also formulate to discover other forms of structural variation that we didn't do yet. A master student is now working on it. We can also, I theoretically find deletions, insertions, duplications, and inverted duplications. Finally, I would like to thank my group at Bilkent University. So the 10x genomics and pooled clone sequencing analysis was largely Marzi's work. Now Fatih is a new master student. He is taking all the hard work. And my other students, 2 of them are actually here, Fatma and Mohammed, in this summer institute. And my collaborators at Cornell, Davis, Indiana, University of Washington, and University of Bari.